continuing with life science and cell biology in a nutshell. We're learning about the bilayers of cells, which are the membranes that surround cells. So these membranes are made out of phospholipid molecules, which we learned about previously. So phospholipid molecules have polarity as well as non-polarity. They're a large molecule with a fatty acid nonpolar tail and a glycerol backbone attached to a phosphate group, which is the phosphate group is usually attached to an alcohol group. So one side of the molecule is polar, the side with the phosphate. So the phosphate group, the alcohol group attached to it, is polar, and the fatty acid tails are nonpolar. Because of the amphipathic or dual polarity of this molecule, the phospholipid molecule, it naturally takes on a shape which is called a micelle, which is a round shape with the one side or the inner side having the nonpolar attributes attracting one another, and the polarity being on the attracting one another as well. So where the water is, you have the polar side. So if a drop of phospholipids is placed in water, they spontaneously form a structure known as the micelle. Hydrophilic heads orient towards the water. Micelles arranged in a spherical form in aqueous solution of the lipid molecules. So the fatty acids attract one another, and the polar head groups attract one another. So a circular shape is formed. In aqueous solution. So in summary, phospholipids, also called phosphatides, are any member of the large class of fat-like phosphorus-containing substances that play important structural and metabolic roles in living cells. The phospholipids with sphingolipids, glycolipids, lipoproteins are called complex lipids, distinguished from simple fats, which are fats and waxes, and other fat-soluble cellular components such as isoprenoids and steroids. The term phosphoglyceride is used by some as a synonym for phospholipid and by others to denote a subgroup of phospholipids. In general, phospholipids are composed of the phosphate group, 2 alcohols, and 1 or 2 fatty acid tails. On one end of the molecule is the phosphate group and an alcohol. So at least one alcohol. This is the polar group with electrical charge attracted to water or hydrophilic. The other end, consisting of fatty acids, is neutral. It's hydrophobic and water insoluble and fat soluble. The amphipathic nature, containing both hydrophobic and hydrophilic groups, makes phospholipids important in membranes, and they form a 2-layer structure called the lipid bilayer, with the polar head facing out on each surface to interact with water, and the neutral tails are driven inward, pointing towards one another. The lipid bilayer is the structural basis of all cell membranes, is nearly impermeable to ions and most polar molecules. Proteins embedded in the phospholipid matrix transport many substances through the membrane that can't get through. So here's a diagram showing the bilayer. You have the outer surface and the outer surface of the cell, the inner surface of the cell. So you may have cholesterol molecules, integral or intrinsic proteins in the membrane, peripheral or extrinsic proteins. So the proteins help with signaling as well as function as channels, helping molecules that are polar or very large and impermeable through the membrane to cross. Um, so you also may have sugar or carbohydrate side chains on the proteins used for signaling, etc., and recognition, such as in receptors and such. So the cell membrane knows what molecules and what it needs to recognize and bind and let through, etc. So, for example, lecithin, which is phosphatidylcholine, and cephalins, phosphatidylethanolamine, and phosphatidylserine, are groups of phospholipids of widespread occurrence in plants and animals. Lecithin is the most abundant, but is rare in microorganisms. Other phospholipids, including plasmalogens present in the brain and the heart, and are apparently limited occurrence in non-animal tissues. Phosphoinositides present in the brain. Cardiolipin is initially isolated from the heart. So depending on the function of the cells and the tissues, you have different consistency of the molecules in the layer of the cell membranes and the phospholipid consistency. Intrinsic proteins penetrate and bind tightly to the lipid bilayer, which is made up largely of phospholipids and cholesterol. In the layer is between 4 and 10 nanometers in thickness. Extrinsic proteins are loosely bound to the hydrophilic or polar surfaces, facing the watery medium inside and outside the cell. Some intrinsic proteins present the sugar or carbohydrate side chains on the cell's outer surface for recognition or receptor functions. The lipid bilayer has been established as the universal basis for cell membrane structure. It can be easily seen by an electron microscope. X-ray diffraction, freeze fracture electron microscopy are used to reveal details of the membrane's organization. The bilayer structure is attributable to the special properties of the lipid molecules, causing them to spontaneously assemble into bilayers, even under simple conditions, like the micelles. So lipids or fatty molecules constitute 50% of the mass of the animal cell membranes, nearly all of the remainder being proteins. Nearly 5 to the 10 to the 6th power of lipid molecules are in a 1 micrometer by 1 micrometer area of lipid bilayer. So about that's a million, right? 5 million lipid molecules. So they're very tiny. Or about 10 to the 9th power lipid molecules are in the plasma membrane of small animal cells. So all the lipid molecules are amphiphilic or amphiphatic, having both hydrophilic and hydrophobic tendency. Phospholipids are the most abundant membrane lipids, polar head groups, not too hydrophobic, hydrocarbon tails, making them up, aka fatty acid tails. Thank you.